Oh, and by the way, this is what the iClicker looks like. I brought it just so if you have not seen one. It just looks like a little remote control. That's what it is. It's white. It says iClicker 2. But like I said, older iClickers, they're just plain iClickers, are fine too. For what we're doing, they'll work. As long as it works. Just make sure it works. They are kind of fragile. Don't drop them because they do break. Anyway, any questions before we get started? These notes are on PowerPoint, but like I have recommended, it's a good idea to take notes. You don't have to write every single thing I say down, but if I'm telling you something that may not be on the slide, or maybe an example, or just maybe a different way of talking about it, you can write those things down, or just write down the topics so you stay kind of organized and you know what was covered that day. So that's what I recommend doing. And usually, in 235, if you've gotten through, if you've got a C or better in 234, you've probably got a pretty good idea of how to study for this class, but it is, 234 is tough. This is a little, maybe a little tougher, maybe. And, so you want to, do what's necessary to succeed in this class, okay? All right, so, and feel free to, come to my office if you have questions about any of the material. If you're not sure, if you need me to explain something again. And before exams, I do offer the opportunity to do study review sessions. So, when that comes up, we'll talk more about that. So, the nervous system. Hopefully you know that the central nervous system and peripheral nervous systems are the major parts of the nervous system. And, let's see here. I'm not used to this screen, so I'm going to have to get adapted to this screen. But anyway, central nervous system, CNS, there's usually abbreviations. When you deal with the nervous system as well as the endocrine system, there's a lot of abbreviations, so you have to get used to that. But CNS is central nervous system. That's the brain and spinal cord. This is the processing center of the nervous system, kind of like the processor on a computer. The communication lines to the outside of the central nervous system are in the peripheral nervous system. Peripheral in this case meaning outside of the central nervous system, and that includes, and that's primarily the nerves that connect the brain and spinal cord with the rest of the body. So cranial nerves in the brain, and spinal cord or spinal nerves connected to the spinal cord. Ganglia. These are little clusters of neuron cell bodies outside the central nervous system. You should have seen ganglia associated with the spinal cord, but also we will see ganglia associated with the autonomic nervous system. But ganglia are defined as being clusters of neuron cell bodies in the peripheral nervous system. Now, the peripheral nervous system also includes sensory receptors and special sense organs. By special senses, we're talking about the sense of vision, hearing, equilibrium, smell, and taste. All those that are there in the head are considered to be the special senses, as opposed to kind of general sensations in the rest of the body, like touch and pressure and pain and things like that. But those are also part of the peripheral nervous system. And we will be talking a great deal about the senses after we cover the autonomic nervous system. This is a kind of schematic of the same thing, central nervous system at the top, and then information going to and from the central nervous system here in the peripheral nervous system. And within the peripheral nervous system, or PNS, outside the central nervous system, you have the somatic nervous system and the autonomic nervous system. What's the difference between those two? Anybody remember? Yeah.
Somatic skeletal muscle, but autonomic skeletal.
Yeah, that's one difference. Yeah. Mm-hmm.
Is one voluntary and one involuntary?
And one's voluntary, one's involuntary. Exactly. So the somatic is the one that you were saying, skeletal muscle. It stimulates skeletal muscle, but it also involves the senses that you are consciously aware of. So somatic is what you're consciously aware of and what you can consciously control. And so the senses, somatic senses would be senses from your skin, for example, like touch, pressure, pain, temperature, heat and cold, things like that. We'll be talking more about those senses later. And also the special senses of vision, hearing, equilibrium, smell, and taste. Those are also somatic senses. And that is transmitted by sensory neurons to the central nervous system. And then in the somatic motor portion of this, motor means that motor neurons stimulate other cells. They stimulate effector organs. And in this case, it's going to control voluntary contractions of skeletal muscle. Now, that being said, some of these contractions or some of this control is at a subconscious level as well, because your different parts of your brain are controlling your muscle contraction. So you have your cerebral cortex controlling your movements. You know, the cerebral cortex is what controls your voluntary movements. But you also have regions of the brain, specifically the cerebellum, back here at the base of your brain, if you remember that, and the basal ganglia deep inside your brain that help in coordinating movements and preventing unwanted movements of skeletal muscle. So they're kind of coordinating those contractions so you're not flailing around and not shaking uncontrollably. So there is some subconscious control of skeletal muscle. Plus, when you maintain posture, there's a certain amount of subconscious control there. You can change your posture, but you don't usually have to think about it all the time. And breathing movements, which involve skeletal muscle, also involves some subconscious control. You have in your brainstem respiratory centers that control breathing movements. So while we say this is all voluntary, there is some involuntary or subconscious sorts of things going on to control and coordinate this. Same thing like when you're blinking, you're swallowing. You're not really thinking about that usually. It's kind of done automatically. And there was, like reflexes. You know, if you burn your hand and pull your hand away, that is, while it's controlled by the somatic system, that's somewhat involuntary too. So there is some subconscious and involuntary function, but it's primarily voluntary. And then you have autonomic, and like you were saying, smooth muscle is one of the things that's controlled by the autonomic nervous system, but also your heart, the cardiac muscle is controlled by this, and glands, both exocrine and endocrine glands are stimulated by the autonomic nervous system. That's the motor portion, and we're going to be talking about that when we get to the autonomic nervous system. The motor portion is divided into the sympathetic and parasympathetic divisions, and that is the two sort of opposite divisions. Sympathetic is your fight or flight response, and that's what controls your body when you're in a state of emergency, but also when you're exercising and things like that. So it's the part that stimulates heart rate and stimulates, breathing faster and things like that. Parasympathetic, one of the names for that is the rest and digest system. So when you're resting and you're digesting a meal, that's what your parasympathetic division controls. So depending on the situation, sometimes your parasympathetic division's in control, sometimes your sympathetic division's in control. They're kind of, modulating back and forth depending on the situation in your body at the time. And both of these divisions are either going to stimulate or inhibit smooth muscle contractions depending on the situation. If it's the sympathetic division, it's going to stimulate your heart. If it's the parasympathetic, it's going to inhibit it, slow down the heart rate. It's going to either stimulate or inhibit glandular secretions depending on the situation. Like your parasympathetic is going to stimulate digestive gland secretions. Sympathetic is going to inhibit digestive gland secretions. So, it depends on which division you're looking at, whether it stimulates or inhibits. Now the autonomic sensory system, this is sensors inside your body, some of them in your internal organs, some of them in blood vessels that are monitoring things like stretching of organs, like stretching of your stomach, stretching of your bladder. It monitors things like chemicals in your food, chemicals in your bloodstream, pH of your bloodstream. Also monitors things like blood pressure, things like that. So these are monitoring internal conditions, most of which you're not aware of. Some of them you are, stretching of the bladder you're aware of, but you're not going to be aware of your blood pressure. You're not consciously aware of it. But you do have monitoring of blood pressure, but that's in that autonomic nervous system. Now, enteric, we're not gonna worry about that one right now. That is part of the digestive system. It's kind of a built-in little system in the digestive tract, and we'll talk a little bit about that when we get to the digestive system. So we're not gonna worry about that one too much right now. Any questions about this picture? Okay, so, make sure I'm keeping an eye on the time. Okay, so first of all, the functions of the nervous system, you should already know, are sensory, integrative, and motor. Sensory is this, detecting sensations, detecting stimuli, either outside or inside the body. Some of the sensations you're not aware of, some of them you are, as I was mentioning before. And this information is transmitted to the central nervous system. The central nervous system, both the brain and spinal cord, are processing this information. The brain is storing some of this information as memory. The brain and spinal cord also sometimes make appropriate decisions about what to do with that information. Mostly the brain, but the spinal cord does in terms of reflexes. There is some control there. And then you have the motor function, which is, stimulation of either skeletal muscle or cardiac muscle, smooth muscle, and glands, depending on which division of the autonomic system you're looking at. We call these things that are stimulated, these things here in the yellow box, we would call these effectors. These are what are stimulated by motor neurons, and so the effector at the end of that scenario there is would be those tissues. Okay, so along with having the different functions, you have different functional neurons. Some neurons are sensory neurons. Sensory neurons, like in this scenario, this guy's pinching the pen and stimulating maybe a pressure receptor deep inside the skin. And that sensor receptor is part of a sensory neuron. And so when you stimulate that receptor, you're going to generate nerve impulses called action potentials. And those action potentials are propagated up the axon. There's the cell body of that neuron. In a ganglion, there's an example of a ganglion there. The cell bodies are clustered together in that ganglion. That axon continues into the spinal cord. Now, this is a nerve right here. This is a spinal nerve. It contains nerve fibers, which are axons, that are transmitting sensory information or motor information, either to or from the nervous system. So that sensory information enters the spinal cord, and this axon continues up through the white matter, through the tracts of the spinal cord, up through ascending tracts to the brain. There you have a synapse between that sensory neuron and a second neuron called an interneuron. Interneurons, which are found in the central nervous system. Sensory neurons are in the peripheral nervous system, but interneurons are in the central nervous system, and it's going to process that information and send that information on to another interneuron deeper in the brain, in the thalamus, and that interneuron is going to send information to the appropriate sensory area in the cerebral cortex. So we'll be talking more about that when we get to sensations. But this is the frontal section, that's the cerebral cortex, that's the gray matter, and there's going to be other interneurons that are going to process that information as well. And you have storage of memory, also retrieval of memory, so you recognize that sensation. So there's going to be a lot going on in the brain processing that information. And then, let's say you're not pinching your pen hard enough. So your brain decides you need to pinch it a little bit harder so you can hold on to that pen better. Then an interneuron will stimulate a motor neuron. This is called an upper motor neuron. This neuron transmits information down through the white matter of the spinal cord. And this upper, where's its letter there? It's hard to see it where you are, but you can see this on your computer screen. The upper motor neuron is then going to stimulate a lower motor neuron to send signals out to the muscle of your hand, and you're going to stimulate these effectors to maybe contract a little more if need be. If you don't want it to contract more, you might inhibit nerve impulses so that you relax that muscle. So it depends on the situation. but motor neurons are actually located, the upper motor neurons are located in the central nervous system, and the lower motor neuron starts in the central nervous system but continues on out into the peripheral nervous system. And we will be talking some about these pathways of information as we go through the sensory and motor material later on. Any questions about this picture? Okay, so that's the functional classification. Here is the typical neuron diagram, the typical neuron anatomy. You have a cell body. That is where, if this were a neuron that integrates information and processes and stores it, this is where that happens. This is where the nucleus is. This is where many of the organelles are located. It's called the cell body, the neuron cell body. But it has, it's usually going to be located in the central nervous system, or it's going to be located in ganglia. So in other words, like this ganglion here, you do have cell bodies there, and ganglia in the peripheral nervous system, but these other cell bodies, you can see cell bodies in the central nervous system. Most neurons are interneurons, okay? Most of the neurons in your body are interneurons, so it makes sense that most neuron cell bodies are going to be in the central nervous system, protected there in the brain and spinal cord. But there are neuron cell bodies in the peripheral nervous system. They're located in these ganglia, like this one here. Attached to the cell body, you have dendrites and axons. These are these extensions of the neuron. The dendrites conduct electrical signals. These are called graded potentials, if you remember that term. These signals travel toward the cell body. And usually the dendrites are short and branched, though we'll see some variation in that, but dendrites are usually short and branched, and in this case there's lots of them. And then you have the axon up there. The axon begins at the axon hillock, and that's where nerve impulses begin. The axon hillock conducts nerve impulses, also known as action potentials, down to the synaptic end bulbs. So the axon, by the way, you can have branches. They're called axon collaterals. So sometimes there's axon collaterals that branch off the main axon. The axons and axon collaterals will branch at the end into axon terminals, which end at the synaptic end bulbs. And sometimes they're called terminal buttons. Some people use that term, especially in psychology. But synaptic end bulbs are where the synapse happens with another cell. It might be another neuron, might be an effector cell. If this is a motor neuron, then it would synapse with either skeletal muscle, smooth muscle, cardiac muscle, or gland. So depends. But the axon is going to end there at the synaptic end bulb. And you always have the, action potential always moves in that direction. It doesn't move in the opposite direction. So that always moves in that direction shown there. Now this picture here is the most common type of neuron. It's called a multipolar neuron. Multipolar neurons have lots of dendrites directly attached to the cell body. One main axon, though you can have axon collaterals. And here they're showing the myelin sheath. Remember, the myelin sheath is a covering created by neuroglial cells that wrap around the axon. The myelin sheath in the peripheral nervous system is created by Schwann cells that literally wrap around and around and around, squeezing the cytoplasm out to the periphery. But this insulates the axon so that you can conduct nerve impulses quicker. In the brain and spinal cord, you have a different type of cell called an oligodendrocyte that wraps around and forms the myelin sheath. So, but in either case, you have this myelin sheath that insulates the axon and helps with faster nerve impulse conduction. So these, showing axons with myelin sheaths around them. Only the axon has a myelin sheath, the dendrites don't. Dendrites do not have a myelin sheath. So here you have that multipolar, meaning many attachments for the dendrites and one attachment for the axon. The trigger zone is the beginning of the axon, and it's also where the axon hillock is in this case. Trigger zone is where action potentials are triggered or begin. And so that's the beginning of the axon. And then the axon here is branching into axon terminals and synaptic end balls. Most neurons are this type. Just like the previous picture, most neurons are multipolar. And that's because most neurons are interneurons and interneurons are multipolar. Also, motor neurons are multipolar. So if you know interneurons and motor neurons are multipolar, you know what most neurons are. Now, the other two neurons are sensory. When we get to some of the special senses, like in the eyes, the retina of the eyes, and the olfactory region of the nose, we'll see this type of sensory neuron is called bipolar. It has two poles. It doesn't mean it's involved with bipolar disorder or anything like that. It just means that you have on one end, you have a dendrite, and at its ends are branches. This is where sensory receptors are located or associated with that. So that's where receptors would be. And then information is conducted down to the cell body, and then the trigger zone is on the opposite side. So it's like you have a north and south pole. The trigger zone is where the action potentials begin down to the synaptic end bulbs there. Most sensory neurons are unipolar. Most are unipolar. They have the cell body here with one little attachment. The dendrites are associated with sensory receptors. And then the trigger zone is very close to that, and that's the beginning of the axon. The rest of this is an axon going all the way down to here. So in this case, the axon begins way up here. It doesn't begin at the cell body. It begins here at the trigger zone, close to the dendrites. Okay, the cell body's off to the side. Most sensory neurons are unipolar. And so if you go back to that picture I showed you before, this initial neuron here, here's its receptor. This would be the dendrite here in this portion of the neuron. And then the axon is going to start somewhere over here, maybe right here where that black arrow is. That's where the action potential is being propagated down the axon. And there's that cell body off to the side. That's unipolar. That's a unipolar neuron. Its cell body is off to the side, just like that picture over here. There's a little connection there, but it's off to the side. So that's a unipolar neuron. And then this neuron, if you look closely, you can see little dendrites sticking out of it. This is multipolar. It's an interneuron. There's another multipolar interneuron. There's a multipolar motor neuron. Here's another multipolar motor neuron. Those motor neurons and interneurons are like that. They are this variety right here. So most of the time what you're going to see are these and these if you look at sensory neurons. But we will see the bipolar ones when we get to the sensory, to certain sensory, or certain special senses, I should say. Any questions about the structural classification? Yes?
Axon collaterals, do they have axon terminals too?
Do they have what now?
Axon terminals?
Yes, yeah, because for instance, if you think about, if I was going to show you this picture here, okay, let's say that motor, this is a motor neuron, okay. I would say this is probably a lower motor neuron because it's got a myelin sheath made of Schwann cells and not oligodendrocytes, so it's got to be peripheral. So this would be a lower motor neuron. And here is an example. This is not showing it structurally, but these two neurons here in this picture, this is from the muscle system, but this is showing you a motor unit. This would be one of those somatic motor, or those somatic motor neuron. That's a bipolar neuron. I mean, a multipolar neuron. Sorry. So it would be this type here, and oops, wait a minute. What they're not showing you are the dendrites. They're just showing you this where the cell body and dendrites would be. The axon runs out into the nerve, and here is one axon collateral, and there's, I guess that would be the main axon there, but there's an axon collateral, there's its axon terminals, and synaptic end bulbs synapsing with one skeletal muscle cell. And then here's one axon branching into axon terminals as well, and synaptic end bulbs. This is one motor unit. It's working as a unit. The motor neuron is stimulating these two muscle cells to contract, so it's working as a unit. It's called a motor unit. And so a motor unit is, a motor neuron and all the muscle fibers it stimulates. So that purple one and these two cells here, these are supposed to be muscle cells. That's one motor unit. And then the green neuron, this is another multipolar motor neuron. These are lower motor neurons too. Its axon branches. There's an axon collateral. There's an axon collateral, and that's probably the main axon. But notice that each axon collateral does branch into axon terminals with their own synaptic end bulbs. So yeah, every axon, if you had, like in motor units, like here, you've only got like 2 or 3 muscle cells per motor unit. There are some motor units where the motor neuron stimulates maybe a couple thousand muscle fibers, like the muscles in your arms and legs, big muscles that are powerful have really big motor units. And so you would have, if you had 2,000 muscle cells being stimulated, that would mean 2,000 axon collaterals going out to those different muscle cells, if that makes any sense. So depending on how many effector cells are stimulated, you're going to have that many branches, going to each of those, and each one's going to end in synaptic end bulbs. That's a good question. So yeah, even though they don't show it in that picture, that axon collateral there is going to end in axon terminals. They just usually show it as one thing, but really, typically you're going to have, especially motor neurons, you can have branches there. And also neurons in the brain. There's a lot of branching of axons, a lot of axon collaterals in the brain as well. Any questions about that? Okay. So when they show these diagrams, oftentimes they don't show all the structures. They don't always show all the dendrites and things. You just kind of have to see it as kind of a schematic diagram. Okay, so let's see what time is it. So we still have a little bit of time. Now we get into the anatomy of it. This is where people sometimes run into trouble, because when you're dealing with it, or the physiology, I should say, when you're dealing with the physiology, then you have to start thinking in terms of almost like nanotechnology level thinking, because you're dealing with cells and cell membranes, and you're dealing with ions, you're dealing with proteins, you're dealing with all this stuff happening at the cellular level, and that's where students have some trouble, because you're trying to imagine stuff that you can't see. So you have to use your imagination, and have to use some abstract thinking to imagine this stuff, and also look at animations when possible to help you understand this. Keeping in mind that diagrams and animations are not reality. They are versions of reality, but they're like little cartoons of reality. What it really looks like is not going to be exactly like these pictures that you look at. But hopefully you remember that in the neuron, and this is also true really for most cells in your body, there is a charge difference across the membrane. There is a charge difference across the plasma membrane. This is the plasma membrane of the cell. This is the phospholipid bilayer. These are those phospholipids with their little phosphate heads pointing toward the outside. The blue is the outside fluid outside the cell. And then these phospholipid phosphate heads are pointing toward the cytosol or cytoplasm, the fluid on the inside of the cell. And at the surface of this cell, the outside surface, if you were to measure the charge difference across the membrane, if you were to take a voltmeter that can measure millivolts, most voltmeters measure in voltage, but this is a millivolt voltmeter. Millivolts are 1,000th of a volt, so we're talking very small voltage. A voltage is a charge difference, like 120 volts. That's the charge difference between the beginning of the circuit and the end of the circuit, and that's what drives, that's what allows you to power that projector or the computer or whatever. 120 volts is a very strong voltage. You would not want it going into your body. But this is a very slight voltage. You can touch that membrane and not feel it. It's not going to be something you would feel. It's a very slight voltage. But it means that there's a voltage difference or a charge difference between the inside and the outside of the membrane. So if you took a voltmeter and you put a micro electrode through the membrane, and then on the other side here you have a reference electrode. So one side is measuring the inside of the membrane surface, the other is measuring the outside membrane surface. This voltmeter would read that as -70 millivolts. Because what that means is the inside is more negative compared to the outside, just slightly more negative than the outside. Now, if it were no difference, if it were completely, the same voltage across the membrane, it would measure 0 millivolts. If it was more positive on the inside than the outside, it would measure +70. So it just means, which side is more negative, which side is more positive. If it's more negative on the inside, it measures it as a -70 millivolts, okay? There's a lot of reasons for that voltage difference. One of them is the sodium-potassium ATPase pump, where you're pumping out 3 sodiums out and 2 potassiums in. And think about it this way: if you have, if you've got 3 sodiums, which are positive on the outside, but only 2 potassiums being pumped in toward the inside, there is a charge difference. This is less positive, this is more positive. Now the voltmeter reads this as being more negative, even though it's positive ions, because there's a difference in the number of ions here. So you have fewer positive ions inside, more positive ions on the outside, because the sodium-potassium pump is pumping out 3 sodiums out, pumping in 2 potassiums in per cycle, and that creates a charge difference that is read by the voltmeter as -70 millivolts, okay? And that can be a little confusing because you see all these positive and you think, well, why is that negative? It's just because there's a difference in number of positive ions. And so, in large part, because of that sodium-potassium pump, cells in your body have a slight, it's not just your neurons, muscle cells, glandular cells, epithelial cells. Most cells in the body have a slight charge difference between the inside and outside because your cells are all doing that sodium-potassium ATPase pump thing. Depends on the cell. Some, if you look at muscle cells, they have a -90 millivolt resting membrane potential. Nerve cells typically have on average about a -70 millivolts. So each cell type is variable in that number, but we're just focusing on nervous neurons right now. Now, because there's a charge difference, we say that it is a polarized membrane, just like a battery is polarized. In a battery, you have a positive end and a negative end, okay? And as long as you've got that polarity, let's say this is I don't know what the voltage on this battery would be. I can't remember what the voltage would be, but whatever that voltage is based on that charge difference between the positive end of the battery and the negative end. When a battery goes dead, it means you basically have a 0 volt difference between the 2 ends of the battery. That's why it's dead, a dead battery. All the ions have gotten mixed together and you no longer have that voltage to drive a current. So you have to have that difference in the ends of the battery, and eventually batteries do, go to 0. But as long as it's got that voltage difference, it can drive. You can use that battery to turn on your flashlight or whatever. But anyway, so we say that's polarized because there's a negative and positive end. Well, this is polarized as well, just a very slight polarity. But that allows neurons to be able to transmit nerve impulses. The fact that it's polarized allows neurons to be able to be stimulated so that they can generate and conduct nerve impulses. If it was 0 millivolts, a neuron would basically not do anything. It wouldn't be able to conduct nerve impulses at all. So it needs that -70 in order to do its job. That's at rest. So we call this a resting membrane potential, meaning there's a potential for something to happen, Nothing's happened at rest, it's just a potential that something can happen. This is really a potential form of energy. We consider this a potential type of potential energy, this resting membrane potential that allows nerve impulse to do its job. So, what happens when you stimulate a neuron? You can get a graded membrane potential. Graded membrane potentials happen when a neuron is stimulated, and it happens in the blue zone there. The dendrites and cell bodies are where you see graded membrane potential. That is a slight change in that potential. In other words, a slight change from -70. That's called a graded membrane potential. There's two kinds of graded potentials that happen in the dendrites and cell body when a neuron's stimulated. It can either be a hyperpolarizing graded potential. Hyper means more, like when someone's hyperactive, they're more active. So hyper means more polarized, meaning more negative on the inside. More negative on the inside. So here's resting membrane potential in millivolts, -70. If you stimulate the neurons so that you get a more negative voltage, that's hyperpolarized. If, however, if you stimulate the neurons so that it becomes less negative, that's depolarizing. That means reduced or less polarity. In other words, less of a difference. Notice you're going up towards zero, right? You're moving towards zero, so you're less polarized. So depolarizing is less polarized, hyperpolarizing is more polarized, more negative, less negative. Now, how do you get those? By opening certain graded or certain protein channels. These are called gated channels, and there's two kinds of gated channels that create these changes in the membrane potential. One of them is the mechanically gated channel. This is one type of way that you can stimulate sensory neurons. We'll see that other sensory neurons, like the taste cells and things like that, are stimulated in different ways. But if it's a neuron that is stimulated by some type of physical stimulation like touch or pressure, so touch or pressure receptors in your skin, also vibration, you do have vibration receptors in your skin as well. Those are stimulated by opening mechanically gated channels in the membrane. And then there's also, what I don't have here, sound, which is also a type of vibration. Sound waves can stimulate these. So sensory receptors in your inner ear that are responding to sound waves are also stimulated by this type of mechanism. In the sensory receptor region of these neurons, in the dendrites, where the sensory receptor region would be of that neuron, you have mechanically gated protein channels that have a gate. When the neuron is at rest, when it's not being stimulated, the gate is closed. Nothing's getting through that gate, okay? And this is again, this is the plasma membrane. These are protein channels. But if you touch, depending on the neuron, each neuron is specific for the type of sensation that stimulates it. But let's say it's a touch receptor just under your epidermis. You're touching your skin, and you can feel that. That means you're opening these mechanically gated channels, and positive ions are flowing in. More positive ions are flowing in. Sodium or calcium ions. Those are positive ions. Remember that sodium is Na+. Well, we've got that up there. Calcium is +2. Those are both positive ions. If you've got a flow of positive ions going into a cell, you've got more positive ions going in. That means it's becoming more positive on the inside, okay? So that would create this wave right here in this graph. If you got more positive ions going in, that wave is going to go up, not down. It's becoming less polarized, more positive on the inside. So you're going to get a depolarizing graded potential in that sensory receptor region. This would also happen if you press down and stimulate those pressure receptors. You're going to get opening of mechanically gated channels. Sound waves hitting your eardrum, and those sound waves are transmitted through the organ of hearing to your little sensory cells. They've got those kind of gated channels. and sodium or calcium is gonna flow in and make it more positive. So that is a depolarizing gradient potential, and depolarizing gradient potentials can stimulate the production of nerve impulses, which then get transmitted down the axon. Oh, no, 1 minute. So here you have two other types of ways cells are stimulated. If it's a neuron that can be stimulated by another neuron, such as an interneuron or motor neuron, they get stimulated by other neurons, right? So they have ligand-gated channels. Ligand, in this case, is a chemical messenger. In this case, we're talking about neurotransmitters, okay? And you can have either excitatory neurotransmitters or inhibitory ones. So we will pick up on Thursday with this and continue talking and hopefully finish this and get into the autonomic system.